Welcome everyone for our afternoon 1:30 session with Dr. Kevin Andrerson, Life's Unprecedented Technology. Dr. Kevin Andrerson has a PhD in microbiology from Kansas State. Oops, is it Kansas State, Kevin?
Yes.
Right, I got the right one, good. After his doctoral work, I know that he had a postdoc fellowship with the NIH, the National Institutes of Health, and I believe in Illinois.
Yes, I did the study at the Department of Microbiology at the University of Illinois. Yes.
Great, excellent. He has taught, I know he taught for a while at Mississippi State University, easy for me to say.
Mhm.
And since 2003 or thereabouts,
Yeah,
he,
thereabouts.
yeah, thereabouts, he has been the laboratory director of the Van Andel Creation Research Center for the Creation Research Society. Their new facilities are on the campus of Arizona Christian University in Glendale, Arizona. Kevin also serves as the Chief Operating Officer for the Creation Research Society, which is the oldest international scientific society dedicated to the investigation of special creation. Kevin is a colleague, a good friend of mine. I've enjoyed his camaraderie now for a number of years. And apparently he has done the same. You might not be able to see this, and he will probably deny this, but this has not been photoshopped. This was actually his own handwriting telling me how much he enjoyed. By the way, I'm glad that Kevin got back. We heard before he's had some password problems. Well, it turns out he's still having little password problems. He did pick a new password that he thought was going to work well. So the computer told him, enter your password. He typed in snowflake. It said, confirm your password. He typed in snowflake. Unfortunately, the computer produced an error and said, your passwords are not alike. Well, Kevin was going to have the better of this, so he finally changed his password to the word incorrect. When I asked him why, he says, so when I forget my password, the computer will tell me, your password is incorrect. Well, now on to some serious scientific investigation. I'm going to stop my share. I'll have Dr. Andrerson share his screen, and we'll get on with his presentation. Dr. Andrerson.
Actually, I like that. I'll probably have to use that incorrect password. So,
I found it helpful myself.
all right, see if we can get this up and going. There we go. Okay, so can you see it fine?
We can see it great.
Very good. Let me take a drink for 1 second. There's kind of, I don't know, several different ways to approach this topic. What I really just want to do is kind of a tour de force of what God has done and how it relates really to what I would call the fingerprint of God, just how he has revealed himself in the creation that he has created, in the creation that we live in. A lot of you obviously are familiar with William Paley and the arguments he used there at the early part of the 19th century, where he argued that if you're walking down a path and you saw a pocket watch just laying on the path, you would immediately assume that somebody lost it, but you'd immediately assume that it was a product of a watchmaker. You would never assume that somehow this pocket watch spontaneously formed and came together right there on the path. Even though you didn't see it being made, even though you didn't see it being dropped on the path, that would be what your previous experience would tell you how that pocket watch came to be laying in the middle of the path. And this really was considered a very strong challenge to those that would deny creation. This was considered a very strong challenge to those that would deny that we are here as an act of creation. Interestingly, this was reported in the Wall Street Journal a few years ago, studies show that by elementary age, children start to invoke an ultimate godlike designer to explain the complexity of the world around them, even children brought up as atheists. See, because it's innate in our thinking. God hardwired us to recognize his fingerprint, to recognize his act. That's why Paley was able to argue that you would immediately think of a watchmaker when you saw a watch. You wouldn't think of it having somehow cobbled together, on its own. And as I said, this was considered very powerful evidence for the act of God until later on in the 19th century, when suddenly their thinking began, in my opinion, to kind of take a back seat, if you will, or to kind of try to give Paley back seat. Okay. In his book, Blind Watchmaker, Richard Dawkins stated that yet the living results of natural selections, because he wants to impart it to natural selection, the living results of natural selection overwhelmingly impress us with the appearance of design as if the master watchmaker, as if by a master watchmaker, impresses with the illusion of design and planning. When it comes to complexity and beauty of design, Paley hardly even began to state the case. Now let's break this down for 2 seconds. Dawkins is stating that the appearance of design is absolutely astonishing, but then he wants to say it is an illusion, because in his opinion, natural selection does it all. And what he states then is, in his Blind Watchmaker, that Darwin's ideas of natural selection and time suddenly now replace the watchmaker. No, indeed, natural selection and time can make a pocket watch. Now you got to recognize the pocket watch analogy by Paley was referring to what was considered a very high piece of precision technology at the time, there in 1802. If he would have been living at the time of computers, he would obviously refer more to a computer laying in the middle of a path, which probably really didn't mean that somebody dropped it there. Probably meant somebody got frustrated and threw it there, but that's another story. Anyway, so Paley's alluding to what to him represented at that time a high level of technology. We now have even higher levels of technology that would fit the example just as well today. Either way, it's interesting that we find that Dawkins, for example, is now saying, no, no, no. Paley's arguments have been replaced. They're not a challenge to we materialists. They're not a challenge to we atheists any longer, because we now know that the watch didn't have to be created. It could have self-formed by a process of natural selection and time. Even though, of course, I've never heard of a watch being made that way, but that's what we're supposed to accept, and that's what's considered the intellectual approach. And that gives us an idea of what we have as intellectuals in this world. So this brings us then to what actually is the situation in the biological world, and are Dawkins' claims correct about we can replace the watchmaker with natural selection and time? First thing we have to recognize is that at the time Darwin was writing, he knew nothing about cellular systems, because nobody did. Nobody had any understanding of metabolism, any understanding of membrane transport systems, of genetic regulation. They knew nothing about any of those things. The assumption was that cellular systems are just simple bags of enzymes. And in this context of very simple cellular systems, then it was very plausible not only to think that they could spontaneously self-assemble, but they could also then very easily manifest into more and more complicated structures. So in this understanding of life, its self-assembly and its ultimate increase in complexity, if you will, seemed very plausible. because life wasn't very complicated. So it didn't take very much for life to form, and it didn't take very much for life to alter, for life to evolve. Simply just didn't, a little assembly required, but not very much. And that's the world we bring into then to look at what's really the situation now in biology. So let's start with the cell, because the cell is the simplest of all life forms. I do not believe viruses are alive. That's a whole nother story. I reject the idea that viruses are alive. I think that's being posted by people who want to try to narrow that gulf between living and non-living. Okay, you start with the cell. That's the simplest of all basic life forms. All right, if we look at a single cell, one of the first things we note is that we have a nucleus with DNA packaged in the nucleus, and the DNA is packaged in structures we call chromosomes. Okay, let's have a very brief look at DNA. If you remember your biology, DNA is a double-stranded molecule. It contains nucleotides, adenine, thymine, cytosine, and guanine. Typically, these are abbreviated simply as ATCG. And that then forms the lettering of the genetic code found on DNA. And the sequence of the A's and the T's and the C's and the G's are like letters on a page. The sequence of the letters of the alphabet determine the language being given on the page, just like the sequence of A's and T's and C's and G's determine the language given by the DNA. Okay, so what we're faced with then is a linear code when we look at just the A's and the T's and the C's and the G's. So they form the letters of the linear code. We also then have, analogous to our language convention, in the genetic language convention, we have words. The words we would call codons, for example. And we have sentences. The sentences we call open reading frames. And we have punctuations. These would involve like promoters and stop codons. And we have paragraphs. These would be genes. So just as we see in like the English language convention, we will see also in the genetic language convention that there is an analogy. They both base on similar conventions, if you will, similar approaches of how the language is conveyed. Because of the understanding of the linear code and how it works, it was assumed then that it'd be just like English in that sense. And so in 2004, or excuse me, in 1992, Walter Gilbert wrote that in reference to the size of the human genome, just the size of the human genome, he says in computer terms, it's actually very little. 3 billion bases of sequence can be put on a single compact disk, and one would be able to pull a CD out of one's pocket and say, here is a human being, it's me. So when you look at it just from the linear code, you'd see, okay, we can just put the linear code of the DNA on a CD. And then by that understanding, this CD represents all the genetic information in a human on a single CD. So once again, you see, with this kind of understanding of genetics and this kind of understanding of biology, cellular systems don't seem to be all that complex. And so it doesn't seem to be all that implausible that life would self-assemble, and the self-assembly then would become more and more complicated as it evolved, because simple systems don't require very much. Here is my challenge though. We're in the 21st century. This is not the 19th century. This is not the 20th century. This is the 21st century. And it is far beyond anything that Darwin understood, far beyond anything even that 20th century scientists understood. So it's time to get into the 21st century. And in doing that, the first thing we have to do is move beyond the linear code, because linear code is just the first code of DNA. DNA is actually a three-dimensional molecule. We've always known a three-dimensional molecule, but we've never understood that the three-dimensionality is vital to the information contained on the DNA. But it's more than a three-dimensional model, because the shape and the orientation of DNA is changing constantly, and that shape and orientation changes the informational content. It changes the code. And so the informational content of DNA is actually a four-dynamic information, because it changes moment by moment within individual cells. So DNA is a three-dimensional molecule read in a four-dimensional dynamic. So now we have a four-dimensional language. Always got amused at the Carl Sagan adapted movie Contact, Carl Sagan book that was adapted movie Contact, where they were so impressed that the UFO people sent down what was ultimately a three-dimensional information code. And that obviously was that these are people who are incredibly smart, incredibly intelligent to do such a code in three dimension. And yet we have to recognize that our DNA is actually in four dimension. So it's beyond the UFO people, which, by the way, amusing thing in that movie as well, when they finally made contact with UFO people had no more understanding what was going on than we do. They had no more answers than we do. So they were pretty useless as far as answering the meanings of life. Might as well just say 42. And if you know what I'm talking about, you understand the reference to that. All right, so we recognized at one time, based on the linear code, that certain regions of DNA contained information, but what was not recognized until we began to understand the three-dimensionality of the DNA is that certain regions of the chromosome will interact with regions of other chromosomes. It's called chromosome kissing. And in so doing, only then when the interaction between the two chromosome segments occurs, do you actually now get the full information content of those regions, and only now is it actually read by the cell. Separated, it doesn't have complete information. But when you put it together, there's the complete information. Now imagine that being cobbled together by trial and error. Changes to the DNA sequence, once considered inconsequential or silent, are now known to alter gene expression, messenger RNA function, protein folding, etc., etc. I put two etceteras on there because obviously the list could keep going on and on. In fact, entire biology classes are talking simply about this. Okay, geneticists are starting to understand that the entire genome is loaded with genetic information. It's not just small sequences scattered here and there. So this is back in 2007 statements. This is 14 years ago, and it's even more so now that these observations of understanding that the entire genome is loaded with information suggests that genomic architecture is not collinear, but is instead interweaved and modular, and that the same genomic sequences are multifunctional. Now consider this: when a language convention is able to impart the same code with multiple meanings, it's reached a level of extraordinary sophistication. This is something that I think Gary could chime in on at some point. This is something computer scientists have been working on really their whole careers, is how to impart multiple meanings to the same code sequence. DNA does it. Okay, epigenetics. If you don't know very much about epigenetics, epigenetics will provide phenotypic changes, that is, physical changes to the organism without altering that DNA sequence. In other words, the sequence of the A's and the T's and the C's and the G's, the sequence of the letters remains the same, but we can alter the effect of that sequence. In other words, we can give it multiple meanings, for example, through the process of epigenetics. There's many mechanisms involved in epigenetics. Methylation of the areas on DNA is one way of doing that. Okay, so here's just a few examples. We can methylate the DNA. We can change the shape of the chromosome. We can modify the structure of the histone protein the DNA is wrapped around. We can acetylate the DNA. We can use small pieces of RNA to silence larger pieces of RNA or to silence the DNA itself. There's just many, many events in epigenetics that will alter the phenotype of the organism but have no effect on the DNA sequence. So this is a whole additional level of genetic information that's not just the DNA structure or the DNA sequence. Okay, now, don't get lost in the technical detail of this diagram. I'm just showing you the intricate mechanisms that are involved in epigenetic regulation within the cell. yes, evolutionists will argue that the most primitive cells would not have had these epigenetic events going on, but we have to understand that epigenetic control is found deep in the biological world. So it's not a latecomer in the evolutionary sense. It had to have occurred early. So even though the first cell may not have had any kind of epigenetic activity, certainly cells soon after began to develop epigenetic events, epigenetic activity, because we find it so deep in the biological world. So it had to have occurred at some point early in evolutionary history, making again the understanding of how these processes and how cellular systems evolved even more formidable than it was before we began to understand epigenetics. So let's look, for example, at the addition or removal of chemical tags to the DNA will alter the chromosome shape. And remember, we mentioned that chromosomes act with a three-dimensional language in a four-dimensional dynamic. So recall that four-dimensionality, because the way in part that four-dimensionality comes into place is by adding or removing chemical tags. But how does the cell know which tag to put or which tag to remove? How'd that come about? Was that cobbled together by trial and error? Let's see the effect of this type of activity on the organism. Let's go with the mouse. Okay, if we have a mouse, a generation of mice, that every time, mice love cherries, and every time they're given a whiff of a cherry, they're given also an electrical shock. After a period of time, the mouse will become very anxious just simply by the smell of the cherry without being given the electrical shock. It's very much a Pavlov's dog type of reaction. But here's where we go beyond Pavlov's dog, because what we find is that the next generation, the offspring of these mice that have been conditioned to the electrical shock, they still react with the same kind of agitation and anxiety every time they whiff the cherry, even though they've never been shocked. And what we find is they have the same epigenetic tagging as their parents developed from the conditioning of the shock and the cherry aroma. So this was passed on to the children as a epigenetic condition, even though the children had never been conditioned in that way. And so what we're finding is that epigenetics not only is something that can influence you or be affected, if you will, by your day-to-day experiences, but it can also influence your children's behavior. What is your epigenetic tagging in your children as well? So Lamarck does come back to us. If you remember, Lamarck argued that, conditions that affect you and can affect you will alter, then also be passed on to your children as well. And that, of course, was rejected, but now we're beginning to see that conditions that begin to alter you do potentially become inheritable. So let's look at some even more fascinating results of what epigenetics does. Take a situation where a small male mouse is placed into a pen with large male mice, and it's traumatizing because the large male mice are very aggressive. And so the traumatized small mouse, then when he has offspring, his mouse remain traumatized small mice. So they have the same physical characteristic as their traumatized small father. They become traumatized and small, even though they have not been exposed to the large aggressive mice. But here's where it really gets strange. Same situation. The small mouse is traumatized by the large mouse. He remains small, he remains traumatized. But instead of reproduction by way of the natural process, if you will, it's artificial insemination. So the female never sees the small traumatized mouse. And what we have then is the offspring or normal size, normal behaviored mice. Apparently this is epigenetic control in the female. And no one has any understanding of how that works. No one has any understanding of what's going on here, but it involves epigenetics. We just don't know how. So this is a level of epigenetics that is so complicated that we don't even have a clue what's going on. And as I said, remember, even though the evolutionists will argue that epigenetic events as we see them today would not have been in the earliest of cells, we recognize that epigenetics goes way deep in the biological world, so it had to have evolved early on. It couldn't have been something that came just in much more recent times. Okay, genetic activity is highly regulated, both by epigenetics and other events. There are numerous regulatory events going on in the cell. Okay, regulatory networks of genes are required for embryonic development of sea urchin skeleton cells. This is the path, if you will. This is the diagram of the regulatory network required for the development of the sea urchin skeletal cells. Now, by comparison, sea urchin skeletal cells would be very simple compared to yours. So your regulatory network would be much more complicated than this one's. And already we look at this and we begin to see just the sophistication of this network. And in fact, and Gary might already be ahead of me on this, we look at this network and we begin to see this looks like a computer logic gate algorithm. This supposedly was just cobbled together by trial and error. In fact, if we look at it, we would see that regulatory networks control gene expression in a way that's analogous to computer programs. Genes grouped into folders, folders grouped into superfolders, chromosomal regions arranged into loops and organized into megafolders. Individual chromosomes interact with regions of other chromosomes. Genes in active folders are expressed in response to various inputs to the cell. Combinations of these folders act like logic gates. The output results in carefully regulated cellular activity. And all of this we're told from the biological community, from the secular biological community, just cobbled itself together over time by trial and error. I don't think Gary writes his computer programs that way. All right, so maybe the best way to look at what we've talked about so far is to go to an analogy where we're going to compare the genetic activity in the cell with how you print War and Peace. The way that your genome functions is somewhat like if we took a 2,000-page volume of War and Peace and printed it on 20 pages. Now, this is not 20 pages of microscopic print. This is 20 pages of normal size print. But what you have then is it's printed in such a way that you would read the page left to right, top to bottom, then turn the page over and read it again left, right, top to bottom. Then take another page and fold it into that page and read page number 3, and then take another page and fold it into that page and read page number 4, and you'd keep folding and turning and folding and turning and folding and turning until you've read the entire 2,000 pages. And it was a complete novel. It wasn't just scattered little stories. It was a complete novel. So the question now becomes, would we even know how to read that book? See, I don't know how to read that book. We would obviously then have no idea how to print that book. This would be beyond, printing such a book would be beyond all the combined intelligence of all humans ever. And yet we would recognize this book has already been printed. It's called our genome. Clear double fingerprint of God. This cannot be cobbled together. You could not cobble together such a printed version of War and Peace. I don't care how many monkeys are typing. I don't care how many universes you're using. I don't care how much time you have. You're not going to be able to put together a book that's going to serve or be read in this kind of manner, because the sophistication, the intricacy, and the intelligence necessary to put that together is just beyond anything that you can argue with or is there anything you can argue by using trial and error, natural selection, time? Just isn't going to do it. Okay, some other examples of innovation in the cells, moving beyond just the genetic code. Let's look, for example, at DNA repair. Now, this is in bacteria. Okay, remember, bacteria supposedly represent the simplest of the life systems that we have on this planet. Okay, this is the strand. This is representing a double strand of DNA. And there is an enzyme called Endo3, and it releases an electron. And this electron flows across the entire strand. And if there's no break, there's no damage to the DNA, then when it hits the enzyme called MUT-Y then releases from the DNA. The function of MUT-Y is that it goes about across the DNA and looks for areas where there's damage, and it repairs the damage. But when it makes contact with that electron, then that electron is in effect telling the MUT-Y, there's no damage in this sector of the DNA because I was able to make it all the way across. And if there was damage, I wouldn't make it all the way across. So this saves the MUT-Y a lot of time. So it can jump off and go somewhere else. And if it encounters an electron again, oh, okay, that electron made it all the way through, so there's no damage in this segment of DNA either. And so what this saves is an enormous amount of time of the repair system scanning areas of DNA that there's no damage on. Now, let's say that there is damage, that there's been a break, for example, in the nucleotide sequence of this strand of DNA. So when the electron is released by the Endo3 and comes to that break point, it stops, it can't flow through. So now the MUT-Y, which is moving on down the DNA from the other side, isn't encountering the electron. So it says, oh, there must be a break down here somewhere. So it keeps moving till it finds the break and it repairs it. So fascinating technology in bacteria, the simplest known life that we have currently on this planet. Also, your chromosomes have an acoustic vibration. In essence, they play music. And this acoustic vibration helps them maintain more of an open structure so they don't get too tangled and knotted up. It'd be fascinating to listen to the music of the chromosome. So literally the music of life. Okay, chromosomal DNA replication. There's over 100 proteins involved in replicating chromosomal DNA. I know when I lecture my genetic students about DNA replication, they usually feel like their brains are ready to explode. It's so complicated. It's so elaborate. It's so involved. So to claim this was somehow cobbled together by a process of trial and error, to me, boggles the mind and shows just how far humans will go to deny their creator. And so when we look at biological systems, we recognize we're looking at a technology that's unrivaled by human engineering. I mean, just think of what we saw with the epigenetics. Just think of what we saw with just the simple electron moving down the strand of DNA. There's so much going on at a biological level that we wish we could duplicate with human engineering. And yet, isn't it fascinating that we're told, oh, these things just cobble together? Do we cobble together human engineering? Hopefully not. Human engineering is a result of millions of man hours, millions of woman hours, millions of people hours of study, of understanding, of experimentation, of trying to understand what's going on. ATPase. ATPase is an enzyme in the cell that's so complex that it can take energy from the flow of protons and use that energy to build ATP, which is the energy molecule or the primary energy molecule in the cell. No one really understands yet fully how this works. We have some basic understanding, but how it ultimately works, no one yet understands. It's that complicated. And even of those that understand most of it, you're talking about probably a number you can count probably on one hand. There's not many people in the entire world that understand most of it. It's just that complicated. And yet again, the idea that somehow this elaborate enzyme cobbled itself together. As proteins flow in the cell, well, as they're formed and flowed in the cell, okay, they're going to move through various compartments in the cell. Now, this is another system that evolutionists will claim was not in the earliest primitive cells, yet these organelle compartment system goes deep into the biological world. So once again, they're faced with this had to have occurred early in evolutionary development. It's not something that was just in the last time period, if we will, of evolutionary development. So even though they'd say, well, the first cell didn't have this elaborate compartmentalization, it goes deep enough into the biological, it goes deep enough into the biological world that we'd have to say it must have evolved early on in biology. And once again, we're looking at an elaborate system that we still don't fully understand, and certainly is something that would defy explanation by a process of trial and error. We saw in my last talk where time is used as a hand wave to explain complexity. And of course, time doesn't give you complexity. Time only gives you, degradation. Time only gives you things wearing out. It doesn't give you complexity. Time is not a mechanism. Time is simply a measurement. Okay, moving those protein molecules from one compartment to another involves another protein called kinesin. Now, or kinesin, sorry, kinesin then moves in a basically kind of like a Star Wars walker, if you will, if you're familiar with those in the Star Wars movie sagas. It moves along a scaffold protein, carrying with it its load, going from one compartment to another. Now, how does this protein know where it's supposed to carry its load? And the answer is that the molecule it's carrying, or the load it's carrying, is tagged with a chemical address. Now, how this protein knows what the chemical address says, we don't know. No one really understands the chemical address. This little protein knows. He's smarter than us in that sense. He knows. We don't fully know, but we do understand that this molecule that this protein is carrying has a chemical address on it, and that tells this protein where to take it to, which compartment to go to. So it's in essence, it's its mailing address. Can you imagine trying to cobble this piece together by trial and error? You'd have him walking off in the wrong direction most all the time, and therefore your cells would be dying all the time. How many cells would you be alive to continue to do the trial and error? Many cells communicate through signal molecules, including bacteria. Now, the bacterial communication system may not be as elaborate, but it certainly is there. It certainly exists. Okay, these molecules initiate a cascade of chemical reactions in the cell, so that molecule A will activate molecule B, which will activate molecule C, which will activate molecule D, and etc. and so forth, until eventually what will happen is the cell will respond through this chemical signaling by either making a specific enzyme or making a specific transport protein. As I say, even bacteria have certain levels of molecular communication that they communicate with each other to get certain set of responses. Once more, I understand the evolutionary community would say that the earliest forms of life did not have this type of communication, but because it goes so deep into the biological world, even bacteria, we would have to challenge that it must have evolved early on, had to be there early in life's history, evolutionarily speaking. So again, they're faced with how in the world do you begin to evolve such an elaborate communication system with all of this cascade effect occurring in the cell by simple trial and error? And once more, I would challenge that chances are, most likely what would happen is the cells would die because they're not reading the communications correctly. They're not getting the right responses correctly, because this is not a one piece at a time. This is essentially back to an all or none. If not all the cascade pieces are in place, nothing happens. So you got to have it all there, and you're really not going to cobble it together one piece at a time, because this cascade doesn't serve any purpose other than the purpose it has today. It's not a, well, it used to do this or used to do that, and then it became more elaborate and added more steps. That's not how it works. Okay, muscle contraction. Just the act of moving a single muscle group involves a highly coordinated action of tens of thousands of muscle cells that are each contracting by pulling along a strand of a long strand of actin protein. And obviously this occurs very, very quickly. Obviously slowed it way down so you can see it occurs very quickly to allow the muscle to contract. Every time then you contract any muscle group, this is what's happening within the muscle cells. And it has to be coordinated. Can you imagine if it wasn't coordinated? You would have no muscle control whatsoever. And that's part of what happens then when you develop certain neurological problems where it's difficult to coordinate muscle contraction. Once more, yes, this would not have been in the earliest of cellular systems, but it goes very deep, certainly into the animal world. And so it had to be something that had to evolve early on in animal evolution. Something perhaps you have not heard of, and that is the personality of single cells. We would assume single cells just being chemical entities, they would react in certain predictable chemical manners, but they don't, especially single-celled microbes. Studies were done with protozoa. Now, some of this work was done 100 years ago, but because it wasn't replicated properly, it was dismissed as, well, that person made a mistake. When it was now more recently replicated properly, they in fact were able to see the same results. And that is, for example, certain species of protozoa, which are single-celled microbes, when they're hit with puffs of beads, they react differently per protozoal cell. Some of the cells will contract and then release from the surface. Some will contract repeatedly but never release, and some will bend and contract and then release, or bend and contract but not release. And lest you think, well, that's still predictable, it's predictable in that if we take the same protozoa and do the same action again, it does the same thing. It'll either contract and release, or it'll continue to contract but not release. If you take another protozoa and put it side by side, one will do the contract and release, the other will do the continual contracting but not releasing. So the protozoa are displaying types of individual behavior that would not be expected based strictly just upon what we would predict in a single-celled organism that's reacting strictly on chemistry. So there seems to be something beyond just the chemistry in these cells that we don't yet understand. Because they're single-celled microbes, that means they go very deep into the biological world, and that means that this type of system would have likely been in place very deep in evolutionary history. So it's not, again, something that was only now having evolved. So refer back to a comment that Albert Riva made back in 2014. Now, he was referring specifically to the genetic code, but I think it applies here as well. While the standard view has always been an oversimplified model that did not account for a multitude of other processes occurring inside the cell, its limitations are today more dramatically apparent than ever. And boy, if that's not the case, as we move into the 21st century, recognize that there's so much we did not understand in the 20th century, so much that was definitely so much not understood in the 19th century. So cellular systems are so much more complicated, so much more that we didn't understand. It's become clear the overall picture is much more complex than was ever understood in the past. That's why I'm saying it's necessary for biologists to start thinking in terms of 21st century when they're talking in terms of evolution. Evolution in many ways is still stuck in the 19th century. and they're having a hard time bringing it forward to 21st century, because when you're faced with some of what I've just briefly explained to you this afternoon, you find yourself in a very difficult position trying to understand how that came about through the process of natural selection and time, how that could have come about. And with the idea that the picture is much more complex and will continue to increase in complexities, we then have to say, okay, is anything in biology ever less complex than previously thought? As we worked our way through biological systems, has anything ever said, oh, that's not near as complicated as we thought it was? I can't think of anything. In fact, when I teach first semester biology students, one of the warnings I give them is that they have to learn a lot more biology than I had to learn. And biology is becoming so much more complicated that what we're able to teach in first semester biology classes compared to the totality of biology is much less. And yet even then, they're gonna have to learn much more than their previous generations had to learn. Fair warning to future biology students, because as we understand more and more of biology, we see that it is more and more complex than we thought it could ever be. And yet, see, because evolutionary biology is still stuck really in the 19th century, they're still wanting to take 19th century ideas and somehow think they explain 21st century understanding. And my direct challenge is they absolutely do not. They don't even come close. Evolutionary ideas fall far short of explaining anything that we understand today. Interesting statement made by Francis Crick, of course, famous for being part of the Crick-Watson team that elucidated the original structure of DNA. He stated that biologists must constantly keep in mind what they see was not designed, but rather evolved. Remember, go back to where we started, where Dawkins said it's an illusion of design, and that children, no matter if they're raised in an atheist home, at one point in time in their life, they have an innate understanding that there is a designer, because that's what explains all of this. And I find it fascinating that a comment such as what Crick is making here, you have to disregard what you are observing, which is the science, and remind yourself that it evolved, which you're not observing, which obviously is not the science. So in other words, you have to dismiss the science, that it obviously was designed, to continue to embrace the non-science, and that is that it evolved. I think a very astute comment made by Michael Behe, of course famous for his Darwin's Black Box book, as far as other books, as well as other books. He stated in an op-ed New York Times writing in 2005 that it's important to keep in mind that it is the profound appearance of design in life that everyone is laboring to explain, not the appearance of natural selection or the appearance of self-organization. In other words, we don't even observe those. We don't observe natural selection doing what evolutionists say it does, or self-organization doing what evolutionists say it does. What we do see is design, and somehow they're working to explain that away. And that's the effort that was being made by the evolutionary community even to this day. They're trying to explain away the fingerprint of God. So, for since the creation of the world, his invisible attributes, that is, his eternal power and divine nature, have been clearly perceived, being understood by what has been made, so that they are without excuse. Of course, that comes from the first chapter of Paul's letter to the Church of Romans. Isn't it interesting how Paul, even 2,000 years ago, through divine inspiration, understood that what's so clear about the nature of God could be understood by just looking at his creation. And of course, we can look in more detail at God's creation than they could then or any generation previous to us. And the fingerprint of God just becomes more and more revealed. The deeper we look, the more we see the fingerprint of God. And the more absurd the ideas become that somehow this cobbled itself together by a matter of trial and error over massive periods of time. In fact, interestingly, the sophistication of creation causes even the noted atheist Richard Dawkins to admit that the creator, if this was created, if everything we see is created, he'd have to be so extraordinary that he's going to be a whole lot bigger and a whole lot more incomprehensible than anything that any theologian of any religion has ever proposed. And of course, that's probably, to me, one of the most profound things Richard Dawkins ever said, because that's absolutely true. The sad thing is that I don't think Richard Dawkins understood what it was he just said. He thinks the incredible creator would be necessary to create what we see, or we can simply replace this incredible creator with natural selection and time. No wonder, see, the Apostle Paul said that they're without excuse. He looks at the beauty and marvel of the created world, says that Paley's watch argument was only beginning to address, and says if a creator made this, that creator must be absolutely incomprehensible. But I'm going to replace the creator with just chance and time, natural selection, somehow selecting through and figuring out what it's supposed to be doing. So it's really just trial and error. As I said, it just is demonstration of how far humans are willing to go to deny the fingerprint of God, to deny that they were created, that what we see was created, that there is a creator. And obviously, of course, the reason they deny that is because they don't like the consequence of there being a creator, because now there suddenly becomes accountability. And with accountability, now they don't want to be accountable. So it really becomes a theological and a philosophical position, certainly not a scientific position. There's not any science really to support their position of trial and error. Everything we see is on the position of creation. But because they choose the philosophical position that there is no creator, then that's where they have to always look then, and unwilling to look at what I would say is the full panorama of scientific information. And with that, I thank you very much.
Excellent. Thank you so much, Kevin. This was outstanding. We have some questions, so we're going to hold your feet to the fire a little bit. Let's begin with epigenetics, kind of a two-parter. Could you give us a straightforward definition of epigenetics?
Well, the simplest definition that I could come up with, and if you talk to someone more specialized in epigenetics, they may be able to do better. But the simplest definition I would come up with is epigenetics is a genetic language above the A's and the T's and the C's and the G's. Because the sequence of the DNA itself is not altered. But epigenetics is able to alter how that sequence is read. So therefore, it's another layer of informational control beyond just the sequence of the DNA.
Great. So contrast for us, how does an evolutionary scientist think of epigenetics versus how a creationary scientist thinks of epigenetics?
I have heard evolutionary geneticists address epigenetics so little that I'm honestly not sure how they address it. I mean, clearly they're going to bring up the same playbook of time, natural selection, trial and error, mutation and such, but there's a group of geneticists or molecular biologists, more specifically, that are known as the third way of evolution. You can look it up, the thirdwayofelvolution.com. They're worth checking out. And here's the reason why. They understand epigenetics. They understand a lot of what I just talked about, and they understand that the classic evolutionary scenarios that you're going to find in the textbooks and you're going to find on Discovery Channel and you're going to find in most of the scientific literature don't work. They don't give you what we see in biology. This group of molecular biologists understand that, but they're also not theists. They are by and large agnostics or atheists. So they reject creation out of hand because, again, the philosophical decision, I don't want there to be a creator. So it's called the third way of evolution because that's what they're searching for. They know that the classic way of evolution is wrong. They don't want creation to be true. So there's got to be another explanation. There's got to be a third way. We don't know what it is, but there's got to be. And so that's what the organization, that's what that group is all about, is we're trying to find that third way. We're trying to find an explanation of how epigenetics would form itself, if you will, would self-assemble. We don't know, we don't have any clue, but we're going to find it. And so I used them as an example of, they have made the understanding that the arguments thrown out there by the secular community don't work. They're not going to explain epigenetics. They're not going to explain a lot of what we just talked about. But they don't want creation to be true. So therefore, they're looking for an explanation that doesn't involve the classic mechanisms were talked about in evolutionary community, doesn't involve a creator. It's got to involve something else. They don't know what that is. It's just, again, an example of the extent that people will go to deny their creator.
Great. Question about the mouse experiment that you cited. Did the traumatized mouse father supply the semen to artificially inseminate the female to produce the larger child?
Yes, that was, I'm sorry if you missed that. Yes, the traumatized mouse, instead of being put in the cage then with a female that, well, in fact, let me go back to it real quick. Make sure, you know, if I didn't understand it, if I didn't explain it correctly or clear enough. Back here real quick. Okay, so we have a traumatized mouse. He's traumatized because he's a runt mouse. He's put in a pen with a large aggressive mouse. He then reproduces the natural way. In other words, he's then put in a pen with a female, they reproduce, but the offspring of that coupling, the offspring of that reproduction are traumatized runt mice. Now, do the same experiment, but this time he's not put in the pen with the female. In other words, it's not the natural way, it's artificial insemination. So the female never sees the traumatized runt mouse. The offspring are normal sized, normal behaviored mice. How? No one has a clue, but it involves epigenetics at some level. We just don't know how, because you see, as let me pose it this way. In our arrogance as humans, because, you know, imagine humans being arrogant. Okay, in our arrogance as humans, probably back in the early 1990s, we were beginning to think that we understood just about all there was to really understand about genetics. Oh, there was a few questions here and a few questions there, but we really didn't, you know, wasn't that, we pretty much had a lot of understanding. And then along comes the Human Genome Project. And we begin to analyze in the sequence not only the human genome, but of the mouse genome and other genomes, and begin then to bring in higher technological development in the ability of analyzing the genome and analyzing what the genome is doing. And we suddenly realize, that's where we begin to realize the three-dimensionality and four-dimensional dynamic, and began to realize that, in fact, we knew so little of how genetics actually worked. It's a little bit like you found King Tut's tomb and you thought this is a marvelous discovery, and you're going through and you're categorizing all of the finds and such until you come to the far corner and you push on the wall and the wall opens up and you go through then a very small hallway and you come to the real treasure. And as far as you can see up and as far as you can see down, as far as you can see across, there's nothing but gold and jewels and valuables. And you realize that you were just on the porch. And now you've found the treasure. In the early 1990s, genetically speaking, we were just on the porch. We're now standing there trying to still grasp the extent with which we're being faced with of actually what's happening biologically. And all we've been able to do is do a few simple experiments like this mouse experiment that reveal how much we don't know yet. And it's only going to continue that way. This is generations worth of research yet to understand what we're being told by the secular community cobble together itself. And then they wonder why I wave my hand and say, you're not even being rational. You're not even being rational.
Next question. Nothing you covered, but maybe you can do this quickly. Sure. What's the best explanation for chromosome 2 fusion linking chimps and humans?
Of course, my colleague Jeff Tompkins at ICR has done a fair amount of sequence research on that. So he would really be the one to contact him at ICR. He can give you much more detail than I can give you. But the simple answer is that the evidence presented that there was a fusion is really based upon the supposition that there had to be a fusion. Because if it's no fusion, then you cannot make the analogy or the analysis of the human and chimpanzee evolution. Even Kenneth Miller admitted that if chromosome 2 in humans was not a result of a fusion, then there could not be common ancestry between chimpanzees and humans, which would say, yeah, there can't be. So in essence, what they did in looking at human chromosome 2 is they determined that this might be a point of fusion. And here's some evidence that might suggest that that was a point of fusion. But the simple fact of the matter is that you can explain that evidence in so many other ways that don't involve two chromosomes fusing together to form human chromosome 2, that for the most part, the idea of there being a fusion is really not a very appropriate interpretation. And so just it's very simply put, like I say, you can contact Jeff Tompkins at ICR to get a much more detail. But what it comes down to is they read a lot into the data and they kind of extrapolated, if you will, beyond what the data really shows. It doesn't give the evidence of a fusion that they would want to claim that it gives. And Kenneth Miller's as bad as anybody in doing that because he's not a geneticist. And when Jeff Tompkins contacted him about his evidence, Kenneth actually backed down some. Well, okay, maybe you're right. Well, by what you just said earlier, Kenneth, if there's no fusion, there's no evolution. He hasn't gone that far.
Very good. Next question. Questioner says, I remember Michael Behe testified in court that led to civil courts agreeing with the secularists that only evolution theory was science to be taught in public schools. So has the complexity discovered since then led to the potential for new court cases?
I would say so. Now, I cannot, Michael Behe states his own mind. I can't give justification of why he said or didn't say one thing or another. This is just Dr. Kevin Andrerson speaking here. But I would say that just based on what I presented very briefly here today, when we remove the design argument from understanding biology, in essence, we have knocked the bottom out of biology. You know, the famous statement that biology only makes sense in light of evolution. And of course, that's bogus because biology makes very clear sense without invoking any concept of evolution as far as universal common descent. But I would say that biology only makes sense in light of design argument. If you don't understand that this is design, then your biology doesn't make much sense. And the design argument, the creation argument, this is where creation and intelligent design would overlap. The design argument would say that there are areas of the genome you need to look at that you didn't look at before, which we're now looking at and realizing, oh gosh, I should have looked at this earlier. That there are events going on in biological systems that are assumed to be just a product of evolution, and therefore they're not looked at, they're not interpreted correctly. And then when you come back and reinterpret them later on, leaving evolution out of the picture, then you recognize, oh, we probably didn't interpret that correctly because we were assuming, you know, X, Y, Z based on our evolutionary assumptions, our evolutionary, you know, lenses, our evolutionary framework. When you throw that out, you recognize, oh, there's a whole different explanation going on. And in fact, that seems to be the more appropriate explanation going on. I'm not going to say that epigenetics would have been discovered earlier had there not been such an evolutionary mindset. But certainly the evolutionary mindset did not speed up the discovery of epigenetics by any second, by even a single minute, because it did not fit what evolution was trying to understand, or the interpretation evolution was giving us in understanding genetic activity in the cell.
Thank you.
So yes, I think when you try to limit science to just strictly, it can only be evolution, what you've done is you've now moved science away from better explanations. And that's not really science. That's a dogma.
Yeah. Good point. Next question. There are many resurrection and instant healing accounts in the Bible. For example, the man who was born blind. Considering this lecture that you've given, which he says is incredible, by the way, how might that reanimation and or healing have occurred on a genetic level?
When you get to heaven, you can ask Christ himself, because obviously that is way beyond anything that we would understand. We simply, first off, it would depend on exactly what the problem was to begin with. Obviously, as scientists, we're working using stem cells. Of course, there's a whole controversy of embryonic versus adult stem cell. We're working at trying to regenerate damaged tissue based on stem cell use and such. But clearly, the kind of instantaneous healings we see that Christ did, or the resurrections that he did, Lazarus being a classic example, we would have that's so far beyond anything that any scientist could explain, that you're literally going to have to talk to Christ himself, because Christ deals at a level of science and engineering that, as you would expect him to. He deals at a level that we're mere infants in comparison. We have no idea how he did that.
Good. Next question. Allowing the evolutionists that life evolved by chance, time, and random processes, how do they explain death? This seems very problematic for them, even if you let them get on first base with a walk, they're not going to get home.
So the question is, how do they explain death?
How do they explain death?
Death, yeah. Yes. Well, that's an interesting question. I guess they would look at death, not unlike how we'd look at death, but they would look at death as that at some point in time, physical systems wear out. Now, the contradiction in that from their arguments would be that they're wanting these biological systems to develop and evolve at the same time they're wearing out. So that becomes kind of a conundrum, because they recognize biological systems wear out, as everything that's material wears out, all the second law of thermodynamics and all that. So death is really a consequence of things beginning to wear out. As you age, you don't make as much you don't have as much stem cell activity, you don't remodulate your bones as much, systems begin to fail, mutations continue to accumulate in certain cells, you may develop cancer. Everybody gets cancer all the time, it's just your immune system typically wipes it out. But at some point in time, if you don't die of anything else, you die of cancer. And so I guess from the evolution perspective, they would use the similar argument without necessarily recognizing, though, that seems to be contradictory to their previous argument that biological systems are evolving and they need to be able to develop and expand all the time, because you have this wearing out occurring at the same time they're supposedly developing and expanding. So it's hard for me to maybe be totally fair in their treatment, because I haven't heard evolutionists be very specific about the evolutionary view of death, other than death is obviously necessary so that the weaker population dies out and the stronger, more evolved population can come in. But the actual biological mechanisms of death, I haven't heard them really address that very much from an evolutionary perspective. So I don't want to be unfair to them, but it does seem to me that it brings in some questions that would seem to be kind of juxtaposition to the arguments we're trying to make of how evolution works, because you would have this degrading process leading to death going on at the same time. So I don't know if that necessarily answered the question adequately. I'm not sure, maybe it may be beyond my intelligence to answer that question adequately. So
I don't think so.
It doesn't probably take too much. I'm well educated beyond my intelligence, so.
Oh, We certainly appreciate this, Dr. Kevin Andrerson. I'm going to stop it now. There are probably, I didn't check the chat. There may be some comments there. I know there's one question we didn't get to if you want to look at those in the Q&A. But I do want to, first of all, thank you very much for bringing all this information and giving us a great amount to think about, and especially relating it to the Creator. So what we're going to do now is we're going to take a 5-minute break so we can get on to our next presentation.